everyone. In today's episode, we're going to talk about beyond genetic engineering. What is synthetic biology really? When you think about synthetic biology, you immediately think about gene editing tools like CRISPR. But there is so much more in this emerging field. In today's episode, we break down the common myth that synthetic biology is all about genetic engineering. Joining me today is our iGEM VIT's wet lab lead, Suhas, and our assistant wet lab lead, Edwin, and myself, Srinidhi. So let's start with the basic doubt. What is the difference between synthetic biology and genetic engineering?
So to start off, it's a common misconception that genetic engineering and synthetic biology is basically the same thing, but it's real. It's very different. I mean, it is like quite similar, but it's not the same thing at the same time. So genetic engineering, to put in simple terms, it's to basically edit a book. Like suppose it's already a written book, you're making small changes in the book. So and synthetic biology is like creating a new book itself. Like it's completely different. So, for example, to give a real world application, so golden rice. So, golden rice was to enrich it with vitamin A. So, in that we produced a new gene itself. While in synthetic biology is creating a new organism itself or new pathway itself in the plant. So for genetic engineering, another example comes to mind is insulin. So, insulin gene into bacteria helps produce insulin for us to extract. So, to put in simple terms again, genetic engineering is basically editing, modifying, inserting and deleting a gene. While synthetic biology is creating a new biological part itself or a new pathway itself in broad terms.
Yeah, I completely agree with that. I feel like genetic engineering is more in a lab completely or completely based on DNA or basically just DNA. But when it comes to synthetic biology, we go more into a complete system or a pathway. We modify a pathway to make a whole new organism in sorts, which is, I think, the fundamental difference between synthetic biology and genetic engineering.
Yes, in synthetic biology, we also need to put in various engineering principles. So we need to design it first, then build it and test it also. But for genetic engineering, you're making an edit to the gene. So you already have the design ready. So you just have to, edit a gene and test it. But synthetic biology, you have to design the circuit, you have to build the circuit from the scratch, and you have to test it. And whatever mistakes are made in it, you have to learn from it in order to improve the circuit.
So to summarize, synthetic biology is this umbrella of biological engineering under which genetic engineering comes, right?
Yeah. Yes. Yeah.
Well, that's very interesting to know. Now that we know about the differences between synthetic biology and genetic engineering, let's move on to how the ways in which we can use synthetic biology practically. So, is synthetic biology all about editing genes? I know we just established that genetic engineering comes under synthetic biology, but is the only way to use it in practical life is to edit genes and make the gene perform a new function or a modified function to give the product that we desire, or is there more tools to synthetic biology?
So synthetic biology is not, as we said before, it's not just editing. You're like designing an entire pathway itself. So for example, like in our project itself, we are designing amberin. So for that, we had to check out the genes that produce our required proteins. So and we have to build the entire protein from scratch. And so we have chosen E. coli as our species. Then we have to start from scratch, like from which molecule will it produce amberin. Then we have to do a literature study, design an entire pathway. Because designing a pathway is just not like putting random genes together. It is actually creating a path. And now, in your pathway, there can be many other sub-reactions as well that you can't predict. But we have to ensure that we mitigate those out. So for that, we have to do literature survey. And as far as synthetic biology goes, we're not just editing. We have to insert a new genes. We have to create a composite pathway. So think of it as like Lego bricks basically. To build a structure, you need multiple bricks. Then only a foundation is laid, and then you're building a proper structure itself. So genetic engineering is just you're editing a block, not the entire thing. So your design would be first creating a structure. Then after that, slow by slow, you'll start building on it with bricks, and that will be your parts. So to put it in simple terms, synthetic biology is like multiple genes, like composition of multiple genes together, creating a genetic circuit.
And even in synthetic biology, it's not exactly necessary that we have to do genetic engineering. There are cases where people have done trials of synthetic biology without actually modifying any genes. You can basically not know what you're modifying and still do a synthetic biology product. That's also there. It's not always just genetic engineering, is what I'm trying to say.
Yes, so synthetic biology, in the starting, it would be without any modification because you're designing a path, like it's entirely new territory itself. After that, after you are familiar with this genetic circuit, then only you can make edits to it. So, like it's basically like a computer code. Without knowing what that code actually does, you can't edit it. So, in simple terms, it's that.
Hmm. That's a very interesting answer. Synthetic biology is used in very many real world applications, like you said, for golden rice. Recently, it's used in medicine as well. There was a hospital in Philadelphia where an infant that was born with a very rare genetic disorder, the baby was named KJ. The doctors cured of his genetic disorder using genetic engineering. So normally when we talk about genetic engineering, we talk about cutting edge technology like CRISPR, which specifically modifies a couple of bases within the genes to make it either normal from being an abnormal gene, or to modify the gene for a different function, or to enhance the function in general. So, what other tools like CRISPR can you use when it comes to gene editing, or is it the only one?
I would say CRISPR is a very powerful tool, but it's not like compared to other tools. There are other genetic tools as well, but CRISPR is mainly just a more powerful tool, I would say. So now, 20, 30 years back, no one would have imagined that CRISPR can do gene editing. Now we are able to cure predisposed genetic diseases. So 20, 30 years back, that would have been something impossible. Now, CRISPR-Cas9 itself was a proper genetic circuit itself. So how it developed, like first they had to design the Cas9 protein to help target the specific sequences. Then after that, you had to edit it out. Then after that, delete that gene. So now a lot of studies are going on to clear Down syndrome itself. Now, 20, 30 years ago, that would have been impossible. But CRISPR is a very powerful tool, actually, genetic tool. That is one of the greatest leaps in synthetic biology and genetic engineering as well.
CRISPR is a very powerful tool, but it's not the only tool. It's just very well studied upon.
Yeah.
It's been perfected, modified multiple times or multiple iterations, and now we have something that actually works very well. But even before CRISPR, we used to do genetic engineering. We used to do it on with raw genes, just using restriction digestion enzymes and ligation, etc., etc. But even now, there are other tools that do the same thing. It's not just CRISPR, it's just that other tools are not so modified. And CRISPR has proven to be more efficient and works better.
Yeah. So some people refer to CRISPR, if I'm not wrong, as precision scissors as well, like for cutting the DNA. So that is as he said, it's a huge leap forward.
Well, technology is constantly evolving, and we should definitely keep looking at the future to develop more and more technologies, especially in the field of medicine, so that we can save more lives. And obviously, as responsible scientists, we have to keep the ethics in mind when we use genetic engineering to modify organisms or to make new medicines, so on and so forth. We will never know where our tools goes to, in whose hand it will go, just in case they might use it for wrongdoing or evil doing. But our responsibility as scientists is first more to enhance the humanity of the earth, as well as protect nature and environment. We should definitely keep our resources safe and develop a better world for all of us. So, when it comes to ethics and values, especially in regards to synthetic biology, where do you think uh, this technology lies?
Um, it's tough to say because again, synthetic biology is a double-edged sword. It can be used for the good side as well and the bad side as well. It's like if you look at our history as well, the atomic bomb was the biggest regret of Robert J. Oppenheimer. So synthetic biology can't really predict what it can lead to. Like as scientists, we can be responsible. We can create it sustainability, like sustainable. We can put it for good use. But at the end of the day, some or the other mindset will lead to its bad. Because now CRISPR-Cas9 is used for genetic editing, but there are a lot of reported cases of it being used for misuse as well. So we can't really predict where it will lead. But as scientists, we should try our best to keep science on the good side and not lead it to misuse as well.
There are always very big concerns on bioweapons being made by multiple countries. There's always a huge possibility that CRISPR could be used to make maybe a virus that could attack everyone. It's a very niche dual use research concern, but it's still very prominent in current world, and that's why. But we can't do anything about it. All we can do is just hope for the best, but it's very possible that it could be used for the wrong.
Yeah, so as scientists, we have to try our best because there are certain factors that we have to correct because not everyone has the luxury to afford these tools, CRISPR-Cas9, etc. Our job is to make it cost effective and reach to a wide variety of public. So as scientists, we have a responsibility to try and cure as many diseases as possible and give them a second chance at life. So as far as CRISPR editing, that's how it started, right? Because certain scientists came with the same mindset that they should be able to cure genetic diseases. Like, why should just this person suffer from it? Like, they should also have a chance at life, right? They should not like end their life at the early stages itself. And as far as the dual edge thing goes, there are a lot of good sides as well. Like we're not considering the fact that we are able to make living sensors that can detect air pollution, water pollution, etc. And the idea of self-healing concrete, self-healing biomaterials. These are all like a very huge leap in technology. Like imagine like 30 years back there was no self-healed concrete. Now that is a huge application in civil engineering. So we should consider always the good sides, and just because there are bad sides as well, like we should not ignore them, but we should try and mitigate them as far as possible.
And there are many people trying to combat these dual-use research concerns. Like some people I've seen have put a kill switch into an organism that when it goes outside its required environment, it just dies. But then again, the people who want to change that could always change it. It's not. It's like hope for the best, prepare for the worst kind of situation.
Yeah, the kill switch part, the soil one, right?
The soil.
Yeah, so
Well, I don't know about you, but personally, I came to synthetic biology to help other humans, like you said, especially in the field of medicine. Well, again, as responsible scientists, we have an ethical board that really scrutinizes our project and makes sure that it doesn't overboard or it doesn't imbalance the natural ecological balance. And despite all of any hypothetical scenarios where this technology would be used for worse, we should always make sure that this research is never less encouraged than it is right now, because synthetic biology will always be the future regardless. And well, it's mostly because we as humans want to make sure that we live for longer. And despite all of that, we also try to make sure that the world is a better place to be in than yesterday. And we only hope to make sure that humanity will progress in the future as we go on. Now, when we talk about the future, where do you think synthetic biology would be like maybe 10 or 15 years?
In 10 15 years, right now only there's a lot of improvement itself. So Right now, I think the research that has been done, like the living sensors and all that, I think they might be perfected hopefully in the 10-15 years. And we soon might have buildings that are made of self healing concrete. So, but at the same time, synthetic biology can yield new ideas as well. So, right now, we have an entirely new generation working on like new ideas. Like in the event we conducted itself, the plasmid event, we can see like they're all working on problem solving and they also have good ideas, but I think some might be limited by the technology at that time, but in future as the technology improves, I feel synthetic biology also will improve at the same time. So I feel in the future synthetic biology will be the one of the pillars and help improving life and providing sustainability as well.
Even now, there are so many really cool advancements. Like, I've heard about designer babies being built. I mean, at least
Test
trying
tube babies?
test tubes babies, and
Yeah.
recently also China made a surrogacy robot where you can completely
Yes, I
just
do have
grow.
heard about that.
Yeah, that's basically inculcating synthetic biology into other fields, combining many, making progress in not just one field, makes it better. And I really do hope, that all of these synthetic biology and every other field like genetic engineering as well would come into much greater use in the future, and seems bright.
Well, listening to all of the applications you just told me, I feel like we're already living in the future. Well, like all the other applications mentioned, we really see how synthetic biology is being used in various advantageous projects, especially at the Plasmid. There was a hackathon that was conducted by IGMVIT where the participants were given the ways in which you could solve real world problems with the help of synthetic biology. And they came up with so many interesting problem statements, like creating an all-in-one allergy testing or adhesive vaccines, so on and so forth, which was very interesting. I found it very interesting. And as demonstrated, we see this synthetic biology being used for the greater good. And that concludes this episode where we discussed about synthetic biology and its applications in genetic engineering. Thank you.